Welcome to the Next Human. My name's Arav, and today I'm here with Jack Prazich. He's a PhD student at the University of Pennsylvania. Thank you so much for being here today, Jack.
Have me, Arav.
Yeah, so let's just get started. So first, one thing I like to ask is like, what's kind of your life story, and like, how did you get to where you are today?
So I grew up in Minnesota, large public high school there, and then did well in school. I just loved math and science growing up. I thought it was always fascinating, and so I went to Santa Clara University first to pursue both an engineering degree and to run Division One, and graduated from University of Wisconsin, and real like I started doing undergrad research at Wisconsin, really liked it, really liked the atmosphere, really loved kind of applying the scientific theory to, you know, get data to prove your point or purposes, and realized I wanted to be a scientist, and realized that scientists you generally have to have to move up, at least in terms of industry. And so I uh, applied to PhD programs straight out of undergrad, got accepted at University of Texas, among other universities. But I loved UT Austin, and then I joined a lab there, Dr. Jung's lab. And during my second year, the lab moved to UPenn. And so I've a little bit, but I'm now PhD candidate at UPenn now going to enter my.
That's great. You mentioned that you fell in love with like the scientific process and the method. But I was wondering like how did you like specifically get interested in cancer research basically?
Good question. So I want to say this for anyone that's watching is you can change the focus of your research at any time. And if you find a field, definitely follow it. So when I was an under, I did research on enzyme, and now I'm doing research on immune system cancer. Very, very, very different. And so that required a lot of learning when I made the jump to the immunology and cancer. But I I find the immune system really fascinating, because it's well known for protecting you from infections, from pathogens, from bacteria to viruses to anything that your body encounters. But it's also incredible that it protects you from it. That's like your own malignant cells if you start to ride out of. And so when I was in grad school, I first did rotations in labs, and that gave me the opportunity to just see what I wanted to. I was most interested in,
Hmm.
and I fell in love with the immune system and cancer. And I think um, a good way to find out what you're passionate about is read publications in that field,
Hmm.
and do they put you to sleep, or are you like, oh, that's actually really um,
Yeah, I think that's nice little tidbit of advice. But yeah, so um I was wondering like what exactly is it that you're like doing? So like what are you doing basically at the lab? And
So I shared a little bit, and if RFB could enable screen, I
yeah,
can
let me do that.
share a little bit about. Like,
There you go. You should be able to do it now. Cool.
do the
Yep, I see it.
okay. So my main research is the T cell response in multiple myeloma.
Mhm.
So multiple myeloma is a form of cancer in the bone marrow. But first, I'll give a little intro on T cells. So T cells and B cells are your main adaptive immune cell types. I focus on T cells. The naive B cells, and those are going to be coming out of your immune system and haven't yet experienced their like cognate antigen. They haven't really been activated. It's the threat. There's regulatory T cells. So these are T cells that are preventing your body from attacking yourself or any self antigens, autoantigens. Uh, and this is also very important for me. And also, and so that they also two main type help cell, and then CD8 T cells. Those are the killing um, any malignant cells or infected cells. And so uh, each T cell has, well, basically every T cell has its own T cell receptor, and that's how it's going to scan for threats in your body. So in your body, uh, tens of, you have billions of cells in your body. Um, you have hundreds of millions of T cells, and because each cell receptor is made up of an alpha and beta chain that are all unique. So there's different B regions, different J regions for the alpha chain. There are different B, D, and J regions for the beta chain, and they can all combine this randomly to create T cell receptors.
Mhm.
So basically, every T cell in your body, unless it is expanded, basically all of the 90 T cells in your body have their own unique T cell receptor, and um, they're basically going to scan for threats, and they're going to scan for intracellular threats or malignant cells. And so how they do that is all the cells in your body basically present uh, this molecule called MHC class 1, or in humans it's called HLA class 1. You might be familiar with it because for, be the match donors, uh, in order for them to not attack any donor tissue as, foreign,
Mhm.
they need to be the match. And so they um, as the recipient. So each person's HLAs are usually very unique to them,
Mhm.
and they will process and present proteins within the cell. They'll chop them up and present them as antigens uh, to the to the immune system. So if a cell gets virally infected, it's going to chop up some of that viral protein, present it as antigen. And if there's a T cell that's specific to that viral antigen, it will recognize it and kill the cell that's been virally infected. Um, that's canonically what CD8 T cells are known for. But T cells in cancer are also really important. So, in your body day to day, uh, you're recycling cells. Some cells die, some cells regenerate. As you get older, it's more and more like DNA damage, other that some of the cells may get malignant. And that means that they are accumulating, cancer is known for cells accumulating mutations in its DNA and eventually full and proliferating. Now, if some of those mutations are then translated into proteins, and those proteins are chopped up in the cell and present a molecule, now those now cells are saying, oh, this doesn't look like a healthy cell. That's a cell, maybe. And, um, so generally, most people have malignant cells in their body throughout their life all the time, but your T cells are constantly removing these threats and stopping them from developing tumors. But as you get older, your immune system is weaker and the tumor develop. And so we know that there are T cells that can still be that still can reside in the tumor and be specific to the tumor, but for whatever reason, on the right here, they can start to express immune checkpoints. They can basically be turned off, become senescent, and no longer cytotoxic.
Mhm.
Um, Many of these T cells will expand to attack, so they'll proliferate, um, attack the tumor and get exhausted and functional, and then they will no longer be able to hold the tumor. And so kind of what I'm doing is looking at the T cell response in multiple myeloma. So multiple myeloma is a malignancy of the bone marrow where your normal plasma cells, which release antibodies, start to divide out of control and lead to a lot of downstream patients. Um, this mainly affects the elderly, and just recently we have started to see a lot of really cool immunotherapy, both um, CAR T cells and immunotherapy drugs. And there are other forms of development. But what I'm looking at is can we use um, as a way to so I'm just going to stop there and you know let you ask any questions. Um,
I mean, yeah.
but yeah, go ahead.
So I mean, this is just kind of like a general question about um, like your lab in general, but like when you're
Hmm.
like looking at like this like response, kind of like how are you guys like doing that, um, in um, like what kind of models and stuff is basically like what I'm asking.
Yeah. So basically, what I've got is blood draws from patients with multiple myeloma, and then I have bone marrow aspirate samples. So they stick a needle in the patient bone marrow, suck up both platelets and cells, and then they, you know, basically enrich for. And so I've got from a clinical trial, I've got blood draws before and after this modulatory treatment and T cells there. And basically, uh, what we use is a multiplexed tetramer panel. Um, so what we do, I'll focus mainly on. I'm just gonna go to the. So basically, how we do this is we take the patient's T cells from the tumor, put it in culture. We take patient's whole exome sequencing. That's basically the expressed genome. Where are their mutations? We predict, okay, of these mutations, which of them are like and which of them could be recognized by the T cells.
Mm.
We then pulse those peptides into the culture with the patient's own basically grow out um, and like make those tumor specific proliferate. And then we use these things called tetramers. So I mentioned how, T cells are specific to a peptide um, that is produced by the patient's HLA. We make tetramers that are basically uh, patient's HLA or presented by the tumor. Got these tetramers that are fluorescently labeled. So when a T cell sticks to the tetramer, sticking to something is then it's short cells, and we can see then we can see what over time and seeing tetramer by what it's specific to.
Well, I mean, it's so actually you're cutting out a little bit. So I think I
Oh, okay.
no no I mean I think I understood most of it. But yeah, I mean, like, first of all, like this is super cool stuff. But if okay, so if I'm right, what you guys are doing is kind of identifying like the antigens that are being like presented by people like with like myeloma through the blood samples, and then kind of like um getting like tumor specific T cells from those like samples.
Yep. So we're
Okay.
seeing what are the patient's what is the patient's T cells targeting on the tumor, and
Mhm.
then what is that T cell receptor, and you know, is it is it proliferating? What's the phenotype of these cells? Like, are they exhausted? Could they be like reinvigorated?
Mhm.
Um,
All right. And then so yeah, so you guys are kind of um uh like seeing kind of like their status basically. They're like okay,
yeah,
cool.
yeah. And so like the next step would be like, okay, if we took this T cell receptor that we found is specific to the tumor,
Mhm.
can we put the patient's tumor in a mouse and transfer T cells with that T cell receptor? Can it kill the tumor in the mouse?
Mm. Wow. That's cool.
Yeah.
Yeah. Um yeah, that's cool.
Yeah. So I can pull out there and
Yeah. I mean, wow, that's like crazy stuff. Um
yeah, I mean, there are difficulties with every project, but I'm doing my best to find tumor-specific T cells.
is it just you?
Yeah, I would say the project is mainly driven by me.
Hmm.
It's basically my thesis project.
Cool.
So if I can complete this, I can graduate with my PhD.
That's great. Are you planning on graduating this year?
My tentative defense date is August 2024. So
Oh,
yeah,
wow.
end of next summer.
Yeah, wow. That's great. But yeah, I mean like that stuff's exciting to me, but I'm sure like to you it must be like even more like exciting and stuff. But yeah.
Yeah, it's exciting when it works. But talking about what I'm doing versus the day-to-day work can look very different.
Yeah,
Yeah.
that makes sense.
Like I said, like predicting, you know, some of like some what are the mutations um, that are like could be a tumor. What that really means is I'm in the office using, you know, bioinformatic tools that other people have developed to make a pipeline, and it's just typing away in a little.
Mhm. Yeah. Um, so yeah, I guess like difficulties in like this whole thing, that'd be be a pretty big one. That I guess it's just easier to like talk about it like theoretically, and then like like the need for like computational tools is pretty heavy in this research, I guess. Right.
Yeah, so I mean, the foundation, like for me to grow out the tumor-specific T cells, I have to correctly predict which of them are presented tumor. If I
Mhm.
don't, then same thing, because I won't be making tetramer specific at epitope, so I may see cells. Um, there are also a lot of epitopes that are probably not presented by the tumor that I'm throwing into the culture that are not doing anything. Um, you know, previous publications have shown like of however many neoantigens you test, you might get 1 to 2% that are positive.
Mhm.
So correctly predicting neoantigens that are is definitely a challenge.
Yeah. Sorry. Um, but yeah, I mean, wow. It's really cool stuff. But yeah, I mean, so that's pretty much it. Um,
Ask you-
what was that?
Can I ask you a question?
Sure. Yeah.
Uh, have you started, do you feel like you want to go into research when you're older? And is there any areas of research that you're
Um, well, I think like research just like in general is just like a such like an important like aspect of like growth in general. Like not just personal growth, but also like, like advancement of the field and stuff. So I think it's definitely something that I want to like get into and actually, try to like do something that has meaning, like, like the work you're doing. So yeah, I mean, I think I definitely want to get involved in research, um, in the future. But and yeah, I think right now, I'm definitely like one area of like definite interest for me is just genetics and just in general. I'm not exactly sure in what like way, shape, or like form that would look like, but the whole field is just like so like interesting to me, and there's just so much cool stuff being done right now that like I think it'd be great if I could like hop into that field. So yeah.
grateful?
Okay, yeah. So I mean, like, what about you? So like next steps kind of after like you graduate and stuff, what do you like, what does that kind of look like for you? What are you thinking?
I think that's been a definite shift in academia for probably 5 to 10 years now. Is many people that graduate are now in the industry through stocks. Um, I saw the value in a PhD though, because many of this, their research team,
Mm.
which I want to continue in, they really want a PhD. During your PhD, don't well. Um, and so uh, I want to be able to. Uh, I think I want to continue in some form of therapy development. Um, and there are clinical trials for child in that area, and then rest.
Oh wait, sorry, you cut out a little bit again.
Ah, shoot.
But I so yeah, could you just like say it again? I got like bits and pieces and stuff, and I think I heard like I definitely want to continue, and then you cut out.
I definitely want to continue in immunotherapy research and development.
Hmm.
In industry, though, I think a lot of companies are focused on the translation of this research into products for patients and for people.
Yeah.
And I want to uh, be able to progress into a management role at some point.
Yeah, I mean that's great. Um, and yeah, I mean I think there's like a lot of um cool stuff going on the industry side too. Um, so yeah, I mean that's great. Yeah. Um, there's one like more question I'd like to ask before we like wrap things up, but it's like what is like one piece of advice you'd give to younger people trying to kind of get into the same field that you're in, and yeah.
Yeah, I think uh, to get into my field, you generally have to um, you need to do decently well enough to get into the program, but after that, it really is more about your problem solving and curiosity and passion,
Mhm.
um, more than your grades. After you get into grad school, your grades do not matter. It is way more um, research, and so a lot of schools now are more focused on the. So if um, as an undergrad, I would reach out to any labs that you find the research people in. Just ask, can I work in a lab? Generally, you're going to start off with the boring work, but um, it's a great experience to then be able to show to that you have always been in research.
Mhm.
Uh, and research looks very different across the spectrum. You could be traditional and you could be in an office all day, or you could be working remote. Um, and on the other hand, you could be, you know, working on a pig farm and testing to see whether, there are the heart uh, a heart valve that you developed works well in a large animal model. So it can research can be very, very difficult. Take your time, figure out what you're and find a guy who is empathetic and wants you to succeed. That is an important part of your PhD as well, is to have a conversation. That's my advice.
That's great. Thank you for that like advice. And thank you again for, agreeing to do this and preparing those slides.
Yeah, thanks for having me, Arav.